Most people think of DNA testing as ethnicity percentages and match lists. But beneath every DNA result is an enormous dataset containing millions of genetic markers. Today we're going to visualize DNA the way a data analyst might see it, not as a family tree, but as structured information that can be measured, mapped, filtered, and compared. DNA is essentially a biological database. Every human carries approximately 3.2 billion base pairs of genetic information. Genetic genealogy companies analyze portions of that information and compare it with other individuals. From a data analytics perspective, DNA is one of the largest datasets most people will ever encounter. Imagine each chromosome as a ruler. Each ruler contains millions of positions where genetic information can be measured. When genealogists compare DNA, they are essentially comparing locations along these rulers to determine whether two people inherited the same segment from a common ancestor. DNA matches are not random. Every shared segment occupies a specific location on a chromosome. Think of it like a street address. We know where the segment begins, where it ends, and how much DNA is shared within that space. This is essentially what DNA matching software does. It compares millions of markers across individuals. When large blocks match, algorithms identify them as potential inherited segments. Those matching segments become the foundation of genetic genealogy. A genetic marker is simply a location on the genome where variation exists. Most people are identical at more than 99% of their DNA. The tiny differences between us become the markers used for genealogy, ethnicity estimates, and health testing. Triangulation is one of the most powerful tools in genetic genealogy. When 3 or more individuals share the same segment at the same chromosomal location and descend from the same ancestral line, we gain strong evidence that the segment originated from a specific ancestor. This is how ancestral DNA can sometimes be identified centuries after that ancestor lived. Chromosome mapping assigns DNA segments to specific ancestral lines. Over time, researchers can determine which portions of their genome came from particular grandparents, great-grandparents, or even more distant ancestors. Ethnicity estimates are not direct measurements of ancestry. Instead, they compare your markers to reference populations. Companies identify statistical similarities and estimate which populations contributed to your DNA. This is why ethnicity results change over time as databases improve. Population bottlenecks occur when a group becomes very small. As generations pass, certain mutations become concentrated within that population. Many ethnicity markers and population signatures exist because of historical bottlenecks and founder effects. These events create identifiable genetic patterns. Mutations occur naturally. Most are harmless. When a mutation survives through many generations, it becomes a genetic breadcrumb. Over time, these breadcrumbs help scientists reconstruct migration patterns and population histories. Modern DNA can reveal connections far older than traditional genealogy. Ancient DNA studies help identify population movements that occurred thousands of years before written records existed. Certain genetic markers influence health. These markers may indicate increased risk for specific conditions or reveal inherited traits. Genetic genealogy focuses primarily on ancestry, but health testing examines many of the same underlying genetic locations. Most genealogy tests analyze selected markers. Whole genome sequencing examines nearly all of an individual's DNA. As costs continue to decrease, whole genome testing is becoming increasingly important for genealogy, medicine, and population research. The future of genetic genealogy will be driven by larger datasets, improved analytical tools, artificial intelligence, and whole genome sequencing. The field is rapidly evolving from simple matching towards sophisticated genomic analysis. DNA is only one piece of the puzzle. A DNA match without records can create questions. Records without DNA can leave uncertainty. The strongest genealogical conclusions come from combining genetic evidence with historical documentation. At its core, genetic genealogy is the process of turning biological data into family history. Each chromosome contains clues. Each segment represents inheritance, and every shared match helps reconstruct the stories of the people who came before us. Thank you for joining me for this exploration of how genetic genealogy works beneath the surface. Understanding DNA as data helps us appreciate both the power and the limitations of genetic testing. As technology advances, our ability to reconstruct family history will continue to improve, but successful genealogy will always require a combination of science, records, and critical analysis. If you enjoyed this video, please like, subscribe, share, and follow for more genealogy and genetic genealogy content from Ancestral Spotlight.